In this video, you will be learning about the skeletal system. This group of organs that include bones, cartilage, tendons, and ligaments is referred to as the framework of the body. However, the skeletal system also performs many other important functions. The skeletal system bears our weight and provides structural support for the soft tissues and attachment points for most skeletal muscles. This cartilage also provides a firm yet flexible support to certain structures in the body, such as the nose and external ear. These strong bands of fibrous connective tissues, which are called ligaments, hold bones together. Another function of the skeletal system is to protect the most important internal organs of the body. For example, cranial bones protect the brain and the rib cage protects the heart and lungs. The skeletal system also helps in generating body movements. This picture shows that there are several skeletal muscles attached to bones. When they contract, they pull on bones to produce movement. Further details will be discussed under our topic on the muscular system. When there is an excessive amount of essential minerals such as calcium and phosphorus in the blood, these are stored temporarily in our bones. If blood levels of these minerals decrease, the minerals are released from bone into the blood. Also, adipose tissues are stored within bone cavities. If needed, the lipids are released into the blood and used by other tissues as a source of energy. Aside from these, the skeletal system plays a major role in blood production. Cavities in our bones, such as the pelvic bones, ribs, sternum, vertebrae, skull, and ends of the bones of the humerus and femur, are filled with red bone marrow, which gives rise to blood cells and platelets. The formation of blood is termed as hematopoiesis. To understand better how these functions are performed, let's look into the structure of the skeletal system. We will first study their microscopic structures. The bone or osseous tissue, as you have previously learned, is a connective tissue. As such, it is composed of bone cells and extracellular matrix. This extracellular matrix, or also known as bone matrix, is a non-living matter produced by these bone cells, which eventually are entrapped within the matrix. The composition of this matrix varies and is responsible for the characteristics of the bone. A mature bone matrix is normally about 35% organic and 65% inorganic material by weight. The organic material includes collagen and proteoglycans, while the inorganic material consists primarily of a calcium phosphate which combines with another mineral salt, calcium hydroxide, to form crystals of hydroxyapatite. The collagen fibers are responsible for the flexibility of the matrix, while the inorganic materials give the matrix hardness or weight-bearing strength. Thus, if we will conduct a simple experiment by submerging a bone in acidic solution or vinegar, which dissolves the inorganic materials in the matrix, the bone becomes flexible since what will remain are collagen fibers only. On the other hand, if collagen fibers are removed, leaving behind the inorganic materials, the bone becomes brittle. There are 4 types of bone cells that are present in bone tissues. These are the osteoprogenitor cells, osteoblasts, osteocytes, and osteoclasts. Osteoprogenitor cells are unspecialized bone cells derived from the mesenchyme. If you recall, mesenchyme is an embryonic tissue or a tissue in a developing embryo from which almost all connective tissues are formed. Of the four types of bone cells, osteoprogenitors are the only cells to undergo cell division. As this cell division occurs, the resulting cells develop into osteoblasts. The osteoblasts are the bone-building cells. Remember that the suffix blast means bud or sprout. These cells are responsible for synthesizing and secreting collagen fibers and other organic components which are needed to build the extracellular matrix of bone tissues. They also initiate calcification or the accumulation of calcium in the bones. As they secrete these components of the extracellular matrix, they are entrapped inside and become osteocytes. These osteocytes from the suffix "cyte", which means cells, are mature bone cells and are the main cells in bone tissues. They maintain their daily metabolism, such as the exchange of nutrients and wastes, with the blood. Like osteoblasts, osteocytes do not undergo cell division. The fourth type of cells are these osteoclasts, which are huge cells derived from the fusion of as many as 50 monocytes. A monocyte is a type of a white blood cell. As seen in this picture, the plasma membrane on this side of the osteoclast that faces the bone surface is deeply folded into a ruffled border. This is where enzymes and acids that digest the protein and mineral components of the extracellular bone matrix are produced. This breakdown of bone extracellular matrix or resorption is essential in the development, maintenance, and repair of bone, as you will learn shortly. These bone cells and the extracellular matrix are not tightly packed in the bone tissue. Rather, there are spaces within that serve as channels for blood vessels that supply bone cells with nutrients. Other spaces also act as storage areas for red bone marrow. The bone tissue may be categorized as compact or spongy, depending on the size and distribution of these spaces. About 80% of the skeleton is compact bone and 20% is spongy bone. As the term implies, a compact bone contains few spaces. It is found beneath the periosteum of all bones and makes up the bulk of the diaphysis of long bones. The periosteum, as you will see later, is this part of the bone that surrounds the bone surface, while diaphysis refers to this bone body. Because the compact bone is dense, it is also the strongest form of bone tissue. Its arrangement along the diaphysis and outside the spongy bone is necessary in providing protection and in resisting stresses produced by weight and movement. As a result, the shaft of a long bone resists bending or fracturing even when considerable force is applied. This picture shows the details of a compact bone. As you can see, the compact bone tissue is composed of repeating structural units called osteons, or also known as Haversian systems. The osteon, which is also the functional unit of compact bone, resembles the growth ring of a tree. The center part, which is known as the Haversian or central canal, contains blood vessels and nerves. It is surrounded by these lamellae that are arranged in concentric or circling layers. The lamellae are circular plates of mineralized extracellular matrix with increasing diameter. Between these concentric lamellae are small spaces called lacunae, meaning little lakes, which contain osteocytes. Radiating outward from the lacunae are these tiny canals called canaliculi that are filled with extracellular fluid. Inside the canaliculi are these mature bone cells or osteocytes. As seen here, the canaliculi connect lacunae with one another and with the central canals. This is important in transporting nutrients and oxygen to reach the osteocytes and for the removal of wastes despite the hardness of the matrix. It is also important in hastening the healing and recovery of injured bones. The other kind of bone tissue is the spongy bone, or also known as trabecular or cancellous bone tissue. Its name implies that it is more spacious or less dense than the compact bone. This feature is important in reducing the overall weight of the bone and in containing the red bone marrow. The spongy bone is located in the interior portion of the bone and are covered by the compact bones for protection. As you can see in this figure, the spongy bones consist of lamellae that are arranged in irregular patterns to form what are called trabeculae. Between these trabeculae are spaces which are visible through the unaided eye. As in compact bone, the trabecula consists of concentric lamellae, osteocytes that lie in the lacunae, and canaliculi that radiate outward from the lacunae. Aside from these small spaces within spongy bone and compact bone, the diaphyses of long bones have this hollow center called the medullary cavity. In spongy bones, the spaces and this medullary cavity are filled with marrow. There are two kinds of marrows, the red and yellow. The red marrow is where blood is formed through a process known as hematopoiesis, while the yellow marrow is mostly adipose tissue. The spaces within bones of fetuses are filled with red marrow. Just before birth, these red marrows start to be converted into yellow marrow. This continues through adulthood. Red marrows in long bones of limbs, except for those in the proximal part of the arm bones and thigh bones, are completely replaced with yellow marrow, while in some bones there are varying proportions of yellow and red marrow. For instance, this part of the hip bone, known as ilium, may contain 50% red marrow and 50% yellow marrow. Hence, in a bone marrow transplant, which is performed to replace deceased or damaged marrow in persons with certain types of blood disorders like leukemia, donated bone marrow is usually acquired here. Also, since it is a large bone, it has more bone marrow than smaller bones and it can be accessed relatively easily. Bones are also classified according to their shape. These long bones, which are usually located in the upper and lower limbs, are longer than they are wide. Flat bones have a relatively thin, flattened shape and are usually curved. Examples of which include certain skull bones, the ribs, the breastbone or sternum, and the shoulder blades or the scapulae. Short bones are round or nearly cube-shaped, as exemplified by the bones of the wrist or the carpal bones and ankle, or the tarsal bones, while irregular bones, such as the vertebra and facial bones, have shapes that do not readily fit into the other three categories. We now study the macroscopic structure of the bones by examining the different parts of a long bone. A typical long bone has the following parts: the diaphysis, which refers to the bone's shaft or body. It is cylindrical and the main portion of the bone. It is composed primarily of compact bone, but it can also contain some spongy bone. The end of a long bone is usually spongy bone with an outer layer of compact bone. The epiphysis is located in the proximal and distal end of a bone. We can see here that between the epiphysis and the diaphysis is the epiphyseal or growth plate, which is a layer of hyaline cartilage. This allows the diaphysis of the bone to grow in length, So that at about ages 14 to 24, when a bone ceases to grow in length, the cartilage in the epiphyseal plate is replaced by bone, and the resulting bony structure is known as the epiphyseal line. Metaphysis is this region found between the diaphysis and epiphysis. This is where the epiphyseal plate or line is located. This part of the epiphysis, where the bone joins with another bone, is covered by a thin layer of hyaline cartilage known as the articular cartilage. This is important in reducing friction and absorbing shock at freely movable joints. Because there are no blood vessels and perichondrium here, repair of damage is limited. The perichondrium is a layer of dense irregular connective tissue that surrounds the cartilage of developing bone. This bone surface that is not covered by the articular cartilage is covered by this periosteum, which is a tough connective tissue sheath and associated blood supply. Some of the cells here enable bone to grow in thickness but not in length. It also protects the bone, assists in fracture repair, helps nourish bone tissues, and serves as an attachment point for ligaments and tendons. As mentioned earlier, this hollow space within the diaphysis is the medullary cavity or marrow cavity that contains fatty yellow bone marrow and numerous blood vessels in adults. It is important in minimizing the weight of the bone by reducing the dense bony material where it is least needed. This thin membrane that lines the medullary cavity is the endosteum that contains osteoblasts and osteoclasts, while this large artery that enters compact bone near the middle of the diaphysis is known as the nutrient artery. Now that we have an idea on the microscopic and macroscopic structures of the bone, we will now learn how bones are formed. The process by which bone forms is called ossification or osteogenesis. Ossification occurs during the initial bone formation in an embryo or fetus. It also occurs as a person's bones grow during infancy, childhood, and adolescence until their adult sizes are reached. Although a person's bones stop growing, ossification occurs during remodeling or replacement of old bone by new bone tissue. Also, ossification is responsible for the repair of fractures or breaks in bones throughout life. Let's first see how bones are formed in the embryo or fetus. By the way, in humans, an embryo refers to the early stages of development from 0 to 8 weeks, while the fetal stage starts from 9 weeks until delivery. At about 5th week of embryonic development, the mesenchyme condenses around the developing brain to form a membrane of connective tissue. There are two different methods of bone development, the intramembranous ossification and endochondral ossification. In intramembranous ossification, bone forms directly within mesenchyme. This begins at approximately the 8th week of embryonic development until 2 years after birth. Most of the bones in the skull, part of the lower jaw or mandible, and the diaphysis of collarbones or clavicle develop through this method. Let us see how intramembranous ossification occurs. Ossification starts in this site of the membrane called centers of ossification. Specific chemical signals will cause the cells of the mesenchyme to cluster together and differentiate into osteoprogenitor first, then into osteoblast. These osteoblasts will then secrete the organic components of the extracellular matrix until they are entrapped by it. As this continues, the centers of ossification will then expand to form a bone. Thus, the centers have the oldest bone and the expanding edges have the youngest bone. These larger spaces between the developing skull bones that have not yet been ossified are called fontanelles or soft spots, which are important as they help the fetal skull pass through the birth canal. These fontanelles will soon harden as they undergo intramembranous ossification. The secretion of extracellular matrix will then stop and the cells, now called osteocytes, lie in the lacunae. Within a few days, calcium and other mineral salts are deposited and the extracellular matrix hardens or calcifies. This process is known as calcification. As the extracellular matrix forms, it develops into trabeculae that fuse with one another to form spongy bones, which will surround the blood vessels. The connective tissues associated with these blood vessels will then differentiate into red bone marrow. As this happens, the mesenchyme will condense at the margin of bone and develop into periosteum. The surface layers of these spongy bones are then replaced with a thin layer of compact bone while the inner layers remain spongy. Majority of the newly formed bones are then remodeled or reformed as they are transformed into their adult size and shape. The other method of ossification or bone formation in the embryo and fetus is endochondral ossification. Take note that the affix chondro relates to a cartilage. Thus, in endochondral ossification, bones develop within hyaline cartilage that develops from the mesenchyme. This cartilage begins to develop at approximately the end of the 4th week of embryonic development. The ossification of some of these cartilages starts at about 8th week of embryonic development. However, in other cartilages, this process might begin as late as 18 to 20 years of age. Bones of the base of the skull, part of the mandible, the epiphyses of the clavicles, and most of the remaining skeletal system develop through this method. This picture summarizes the sequence of events in endochondral ossification. Just as in intramembranous ossification, cells in the mesenchyme will crowd together at the site where the bone is going to form. They will cluster together to form the general shape of the future bone. These cells will then differentiate into specialized kinds of cells known as chondroblasts. Like the osteoblasts, these chondroblasts will also produce the extracellular matrix of what will form into cartilage model. The cartilage model will then be covered by this layer of connective tissues known as perichondrium. As the chondroblasts become deeply buried in the cartilage extracellular matrix, they are called chondrocytes. The cartilage model then grows in length by continual cell division of these chondrocytes accompanied by further secretion of extracellular matrix. This type of cartilaginous growth is called interstitial growth or growth from within. The cartilage also grows in thickness, which is due mainly to the deposition of extracellular matrix material on the cartilage surface by new chondroblasts that develop from the perichondrium. This process is called appositional or exogenous growth. The cartilage model continues to grow, and the chondrocytes in its center increase in size, and the surrounding extracellular matrix begins to calcify. Other chondrocytes within the calcifying cartilage die because nutrients can no longer diffuse quickly enough through the extracellular matrix. As these chondrocytes die, the spaces left behind by dead chondrocytes merge into small cavities called lacunae. A nutrient artery will then penetrate the perichondrium, and the calcifying cartilage model stimulates osteoprogenitor cells in the perichondrium to differentiate into osteoblasts. Once the perichondrium starts to form bone, it is known as the periosteum. Osteoblasts then begin to deposit bone extracellular matrix over the remnants of calcified cartilage, forming spongy bone trabeculae. This site, where most of the cartilage are replaced by bone tissues, is known as the primary ossification center. As the primary ossification center grows towards the ends of the bone, osteoclasts break down some of the newly formed spongy bone trabeculae. This activity leaves a cavity known as the medullary or marrow cavity in the diaphysis of bone shaft. Eventually, most of the wall of the diaphysis is replaced by compact bone. When branches of this epiphyseal artery enter the epiphysis, secondary ossification centers develop, which usually happens around the time of birth. The events that happen during secondary ossification is similar with what occurred in primary ossification. The only difference is that in secondary ossification, spongy bones remain in the interior part of the epiphysis so that no medullary cavities are formed here. The hyaline cartilage that covers the epiphysis will then become the articular cartilage. Before adulthood, this hyaline cartilage remains between the diaphysis and epiphysis as the epiphyseal or growth plate. We will now examine how a bone grows in length and thickness. But before this, let us first look into the details of the epiphyseal plate. The epiphyseal or growth plate is a layer of hyaline cartilage in the metaphysis or between this epiphysis and diaphysis of a growing bone. It consists of 4 zones. This layer, which is nearest to the epiphysis, is known as the zone of resting cartilage. The cells here do not function in bone growth, but they anchor the epiphyseal plate to the epiphysis of the bone. This next layer is the zone of proliferating cartilage. Cells in this zone replace those that die at this side of the epiphyseal plate. The zone of hypertrophic cartilage consists of these large, maturing chondrocytes that are arranged in columns, and this zone of calcified cartilage is only a few cells thick and consists mostly of dead chondrocytes because the extracellular matrix around them has calcified. These calcified cartilages are then dissolved by the osteoclasts. The osteoblasts and capillaries from the diaphysis will then invade the area, and the calcified cartilage will be replaced by the extracellular matrix produced by the osteoblasts. As a result, the zone of calcified cartilage will become the new diaphysis that is firmly cemented to the rest of the diaphysis of the bone. Growth in length occurs as a result of interstitial growth of cartilage on this side of the epiphyseal plate or through the replacement of cartilage on this side of the bone. After adolescence, or at about age 18 in females and 21 in males, these epiphyseal plates close, which means that the epiphyseal cartilage cells stop dividing and bone replaces all remaining cartilage. The epiphyseal plate will fade, which results into this bony structure known as the epiphyseal line. Thus, the appearance of the epiphyseal line indicates that the growth in length stops completely. Understanding of this is important in determining bone age and in predicting adult height and age at death of skeletal remains. For instance, if the epiphyseal plate is open, it indicates a younger person, while a partially or completely closed epiphyseal plate indicates an older person. Take note also that the closure of this epiphyseal line happens 1 to 2 years earlier in females. While the bone is increasing in length, it also increases in diameter. In fact, Growth in diameter or appositional growth occurs even after growth in length ceases. Appositional growth, or the increase in the diameter or width of bones, occur beneath this periosteum. This picture summarizes the sequence of events in appositional growth. During puberty, when a bone rapidly grows in width, osteoblasts from the periosteum lay down bone to form a series of ridges with grooves between them. The grooves will then become tunnel that encloses the blood vessel. The name of the periosteum in the tunnel changes to endosteum because it now lines an internal bone surface. Osteoblasts in the endosteum will then deposit extracellular matrix, forming new concentric lamellae. The formation of additional concentric lamellae proceeds inward. In this way, the tunnel fills in and a new osteon is created. As an osteon is forming, osteoblasts under the periosteum deposit new circumferential lamellae, further increasing the thickness of the bone. Bone remodeling refers to the replacement of old bone tissues by new ones for various reasons such as renewed bone strength, removal of injured bone, and alteration of bone shape for proper support based on the stress patterns experienced during the remodeling process. This bone remodeling is made possible by the bone-destroying cells, or the osteoclasts, and bone-building cells, or osteoblasts. During the process, the osteoclast attaches tightly to the bone surface at the endosteum or periosteum and forms a leak-proof seal at the edges of this ruffled border. It then releases enzymes and several acids into the sealed pocket. The enzymes will then digest collagen fibers and other organic substances, while the acids dissolve the bone minerals. This removal of minerals and collagen fibers from bone by osteoclasts is termed bone resorption. Once resorption is completed, osteoclasts depart and bone deposition, or the addition of minerals and collagen fibers to bone by osteoblasts, occur to rebuild the bone in that area. As mentioned, bone remodeling is important in the repair of injured or fractured bone. A fracture refers to any break in the bone. As you can see in this figure, the repair of fracture proceeds from the reactive phase to reparative phase, or the formation of a callus, and remodeling. Several factors influence bone growth and remodeling. Certain vitamins are important to bone growth. Vitamin D, which may be synthesized or ingested, is necessary for the normal absorption of calcium from the intestines, while vitamin C is necessary for collagen synthesis by osteoblasts, and vitamin A stimulates the activity of osteoblasts. Minerals like calcium and phosphorus are also important components of bone and are thus needed in large amounts, especially during bone growth and remodeling. Hormones are equally important in bone growth. For instance, insulin-like growth factors, or IGFs, which are produced by the liver and bone tissues, stimulate osteoblasts, promote cell division at the epiphyseal plate and in the periosteum, and enhance synthesis of the proteins needed to build new bone. Thyroid hormones T3 and T4 from the thyroid gland also promotes bone growth by stimulating osteoblasts, and insulin from the pancreas promotes bone growth by increasing the synthesis of bone proteins. Sex hormones cause a dramatic effect on bone growth. This includes estrogens produced by the ovaries and androgens such as testosterone that are produced by the testes. Although females have much higher levels of estrogens and males have higher levels of androgens, females also have lower levels of androgens and males have low levels of estrogens. The adrenal glands of both sexes produce androgens, and other tissues such as adipose tissue can convert androgens to estrogens. These hormones are responsible for increased osteoblast activity, synthesis of bone extracellular matrix, the sudden growth spurt that occurs during the teenage years. Estrogens also promote changes in the skeleton that are typical of females, such as widening of the pelvis. Ultimately, sex hormones, especially estrogens, in both males and females, shut down growth at epiphyseal or growth plates, causing elongation of the bones to cease. Finally, lengthwise growth of bones typically ends earlier in females than in males due to their higher levels of estrogens.